Hi everyone, today we're going to explore a new generative chemistry tool called VeGA, short for Versatile Generative Architecture. It's a lightweight transformer model designed to create novel bioactive molecules. Let's see how it works, what makes it unique, and how we can use it in drug discovery projects. In drug discovery, we may want to use de novo molecular generation to design new compounds, somewhat from scratch. Modern AI models learn the chemical language of molecules, for instance encoded as smile strings, and generate new structures with crude drug-like properties. These models include recurrent neural networks, variational autoencoders, generative adversarial networks, and transformers, which can predict each atom or bond step by step to form valid, realistic molecules. In the VeGA article, they mentioned that some of the best-known generative models include RangVent, which combines high-performance RNNs with transformer mechanisms, and S4, a structured state space sequence model that captures long-range dependencies in sequences. VeGA builds on these ideas but simplifies the architecture. It's a decoder. Only transformer, meaning it's faster, lighter, and was found to be highly accurate. Despite having under 1 million parameters, it competes with much larger models like MolGPT, which has around 8.6 million parameters, or Tracer, which goes up to 45 million. This shows just how efficient VeGA's architecture is, compact but still very powerful. VeGA is first trained on the Chem database, learning general chemical rules from over 1 million molecules. Then, it can be fine-tuned on a small set of compounds that are active against a specific target. This transfer learning step allows it to generate novel molecules tailored to your protein. However, you still need to have around 100 active molecules or more for an efficient fine-tuning process. In many real-world projects, that's not easy, but there might be ways to work around this. I tested VeGA on factor 10, a key enzyme in the blood clotting cascade. I collected bioactive compounds from the Chem database, fine-tuned the model on a dataset of about 150 molecules, and then used it to generate new candidate molecules. I used a diverse set of compounds as input for fine-tuning, but it could be also interesting to focus on 2 or 3 chemical clusters with many members. For a well-studied protein like factor 10, this kind of experiment is quite accessible since plenty of data are available. But for less characterized proteins, fine-tuning becomes more challenging due to the limited amount of training data. During fine-tuning, I mostly used the default parameters, only adjusting the validation ratio to 0.2. The training ran smoothly, making it easy to test different configurations. Using the best fine-tuned model from the previous step, I generated over 2,000 new compounds. This was just a small-scale test to explore what the tool can do. I then docked these molecules using Data Warrior for a quick and simple screening. About 400 compounds showed better predicted binding scores than rivaroxaban, the approved factor 10 inhibitor. This result gives a clear idea of the potential of VeGA. To wrap up, VeGA is a fast, efficient, and open-source transformer model for molecular generation. It's available on GitHub, and although developed for Linux, I managed to run it easily on Mac OS with a few small script adjustments. If you're working on drug design and want to explore generative chemistry, VeGA is definitely worth trying. I hope this video was useful for your project. Thanks for watching, and until next time, take care.